Shaitanirrajim. Bismillahirrahmanirrahim. Hello everyone. Uh, you are watching me as Zahur Islam on my official channel. Uh, so uh, Today my lecture is on pharmaceutical technology. So, in this lecture, we will discuss about the pharmaceutical nanotechnology, its introductions, definition of nanoparticles, preparation, characterization, drug loading, incorporation of water-soluble drugs, incorporation of 5-fluorouracils into nanoparticle drug release and conclusion. So come toward the basic introduction of the pharmaceutical nanotechnology. So the dream of delivering a pharmaceutically active molecule to a specific site in the body has been a long-held aspiration with beginning that may be traced back to the Fall Enlex, who in the early 20th century coined the phrase magic bullet to describe such an entity. Today, extensive pharmaceutical research has led to the development of drug delivery system and strategies which go some way to fulfilling these ideas, but few which could be described as a magic bullets. Side effects and toxicity still affect these approaches and hence enriched visionary thinking has not yet been fully realized. As we know that there are different drug delivery systems. So one of the most prominent way is how to deliver a drug to the target site. So nowadays, scientists they are working on different areas. So one of the most tremendous area is nanotechnology. So the early to visionary thinking has not yet been fully realized. This is especially relevant in tumor chemotherapy, where selective delivery to the neoplastic cell is in comparison to the surrounding normal cells is an important principle. As it can be seen in the figure 1 that the challenge faced in the site-specific delivery of drug is immense due to numerous obstacles barricading the drug along with the desired route. Cellular structures and indeed the very common components of the cell itself will either prevent or act in some selective manner to hinder the migration of the drug from its point of administration to the intended destination site. On moving forward into the 21st century, it is apparent that modern medicine still faces many challenges. Nanotechnology is indeed one area that may offer scientific advances in the coming years, which could lead to significant process in the improvement of therapeutic outcomes. So, in particular, the development of nanoparticulate drug delivery system may enhance the probability of getting a drug to its target sites. So this is one of the most prominent method through which we can deliver a drug to the specific to the targeted area instead of relying on the physicochemical property of the drug to dictate into the bio distribution. The drug is incorporated as a payload into a particle resulting in a different transit mechanism for the drug after administrations. This can be enhanced further by the virtue of the flexible nature of the nanoparticle scaffold onto which the subsection may be chemically bolted, producing a tailor-made and multifaceted devices. This can be represented in figure 1b, which gives to the blueprint of the idealized nanoparticulate delivery system that must make it the way to the target cells. The particles in figure 1B have several properties which are incorporated into the particle, mostly by the covalent bonding to the surface groups. A targeting system such as monoclonal antibodies, and these monoclonal antibodies will recognize the binding sites that are unique to the target cells and allow the particle to dock onto the exposed surface. This has been the focus of much current research into developing strategies for targeting nanoparticles to the site of action. A fusion proteins will instigate the process of merging with the target cell, thereby bringing the particle into the cytoplasm. As a polymeric nanoparticles, they are recognized as a foreign by the body immune system. They are removed quite effectively by phagocytosis and exposure to the endoreticular system, and this will prevent the particle from reaching the target site and must be prevented. Steps toward this goal have already been taken with the production of so-called stealth nanoparticles. And these are the nanoparticles which incorporate a biomimetic polymers, usually polyethylene glycol into their structure to avoid the elicitation of the immune response. Presently, such an idealized nanoparticle with these three important properties has yet to be realized, but the attempts have been made to attach some of the subsystems described. The construction of an idealized particle must start with the formation of the solid polymeric sphere with the diameters of approximately 200 nanometers. Now you can see here, this is the intestinal wall, and these are the connective tissues, while this one is the vascular endothelium, and these are the targeted cells. So after oral administration, the drug will must be metabolized in the liver before it's reaching to its systemic circulation, and then it will. This is intravenous administration, and this is oral administrations, and these are the connective tissue, and these are the intestinal wall. These are the target cells. These are the vascular endotheliums. You can see here in another sketch the particle permeation process. This is the rate controlling membrane. This is the drug payload, and this one is the rate controlling membrane. This is the targeting system. So here there is preventer of phagocytosis, and here is the fusion of molecules can take place. You can see in the figure, it is a very simplified overview depicting the barriers to drug localization at a target cell after oral administration into the gut or intravenous administration to the systemic circulation. For example, the path from a capillary to a target cell may involve crossing the vascular endothelium and various connective tissue structures, and idealized draught of the multifaceted nanoparticulate system containing a drug payload that must also permeate in some way to target cells. Pendant molecules such as targeting ligand drugs add to functionality of the devices. So as I was discussing about the constructions of an idealized particles which must start with the formation of the solid polymeric sphere with the diameters of approximately 200 nanometers. So this is followed closely by the loading the drug into the necked particles. This is dependent on the physicochemical property of the drug molecule, which may often present the problem in the term of efficient loadings, and especially if the drug has appreciable water solubility. This scenario may not be uncommon given that the majority of the candidate molecules from the inclusions in the nanoparticulate system it will possess some degree of the aqueous solubility. This chapter discusses the construction of the nanoparticle, describe their characterizations. So in particular, the process of incorporating water-soluble payloads is discussed and the problem associated with this are used to illustrate the complexities encountered during the proposed construction of a device is simply you can see in the figure 1b. The construction of the devices are here. The biodegradable polymers they are also used include the polyalkyl cyanoacrylates, while polylactides, coglycolides, and chitosans, gelatins, while the non-biodegradable polymers include polystyrenes and polyacrylamides and polymethyl methacrylates. Nanospheres represent the simplest carrier and they are solid monolithic system in which the drug is dissolved or entrapped throughout the particle matrix. Alternatively, so it may be absorbed onto the surface and no continuous membrane surround the particles is illustrated in the figure 2a. So nanocapsules are a reservoir type system comprising an liquids core surrounded by a polymeric shell and the drug is usually dissolved in this liquid core but may be more closely associated with the shell polymer and exposed surface, which is also shown in the figure 2b. Liposomes are closely related to nanocapsules and the structure's layout, but consist of an aqueous core surrounded by a bilayer membrane composed of lipid molecules such as phospholipids, is illustrated in figure 2c. The drug can be located in the aqueous core and the bilayer membrane. Now you can see here, these are the drug molecules. So the drug disperse in Ily core. The drug is dispersed in the Ily core. While here the drug is absorbed on the surface or dispersed in the shell. It means that these the drugs particles, so they are dispersed dispersed in the shell or these drugs are adsorbed. So when these drugs are adsorption, that is also called surface adsorption. So I mean that these drugs they are adsorbed on the surface. In this case, the drug is dispersed in a particulates in a particle. So the drug is dispersed in the particle while here the drug is adsorbed on the surface. Both are the drug molecules. So in this diagram, you can see this is a drug molecules. In this drug molecule, so this portion or this is called the aqueous core, hydrophilic drug and aqueous core. So the hydrophilic drug, they are dispersed or they are incorporated in the aqueous core. While here in this case, this is hydrophobic. These are hydrophobic drug, which is lipid bilayers, water hating. So here the hydrophobic drugs is sandwich or it is embedded incorporated in the lipid bilayer. In this case, the hydrophilic drug is dispersed in aqueous core. So this there is a major difference. This is hydrophilic drug and this one is hydrophobic drug. Here the hydrophobic drug is sent which are incorporated in the lipid bilayer. And you know that lipid bilayer, there are two layers. There are two lipophilic layers. One is hydrophilic and the second one is lipophilic. If you can see the figure number 2, the morphology of the nanosphere where the drug can be either dispersed throughout the polymeric matrix of the particles or adsorbed onto the surface, while the morphology of nanocapsule where the drug is dispersed mostly in a liquid core. So drug can be associated with the polymeric shell either by dispersals through the polymers or adsorptions to it. The morphology of the small single lamellar liposome of the sub-micrometer dimension showing the possible locations of the drug payload. Now come toward the definition of the nanoparticles. Nanoparticles can be defined as we know that nanoparticles have been studied extensively as a carrier for drugs implied in a widely variety of routes of administration, including the parental, ocular, and perioral pathway. The term nanoparticle is a collective name for any colloidal carrier of submicrons or submicrometer dimension, which include the nanosphere, nanocapsule, and liposomes. They can all are the defined as a solid carrier approximately spherical, and the rangings are in the size from 10 to 1000 nanometers. They are generally nanoparticles. They are generally polymerics in nature. It may be synthetic in nature or naturals, and it can be biodegradable and non-biodegradable. I have already discussed in detail. The biodegradable polymers are given here. You can see polyalkyl cyanoacrylates, and this one is polylactide co-glycolides and chitosan and gelatin. These are biodegradable polymers. Mean these polymers are completely compatible with our body system, while the non-biodegradable polymers include polystyrene, styrene, and polyacrylamides and polymethyl methacrylates. So the nanospheres represent the simplest carrier, and they are the solid monolithic system where the drug is dissolved or entrapped throughout the matrix, the particle matrix. So alternatively, it may be adsorbed onto the surface and no continuous membrane surrounded by the particles. From a historical perspective, liposomes have been the most extensively studied nanoparticulate carrier. However, their full development leading to extensive clinical uses have been restricted by the pharmaceutical problems entailing the drug leakage, the drug stability, and problem and scale-up procedure. I will also proceed my discussions on scale-up procedures. These all arise due to their lipid and semi-solid nature, leading to the lack of rigidity and subsequent fragility. However, the important early work by the Correr and co-worker introduced the nanoparticles prepared from the biodegradable polymers, that is polyalkyl cyanoacrylates. And this has been prompted much research into the nanoparticle made from the polymeric materials, and furthermore into finding polymers that are pharmaceutically acceptable, biodegradable, and non-toxic. A wide variety of preparation of methods for the nanoparticles have now evolved, which encapsulate many different types of drug compounds. Come toward the preparations of nanoparticles and nanospheres. So first of all, as we know that both the nanospheres and nanocapsules, they are prepared from either a polymerization reactions of dispersed monomers or from a soluble dispersion procedure using performed polymers. In many instances, the latter procedures using preformed polymers as desirable as potential reactions between drug and monomers. They are avoided, and the potential toxicity of residual monomers, surfactants, and initiators is reduced. The final properties of nanoparticles, such as their size, their morphology, the drug loading capacity, release characteristics, and biodistributions, and are all influenced by the method of preparations. Nanoparticles, they are prepared from polymerization reaction or polymerization process. Most examples of polymerization used to create the nanoparticles occur by a pre-radicals mechanism involving the distinct initiation, propagations, and termination process. Polymerization occurs within a continuous liquid mediums, which also comprises the monomers, initiators, and surfactants. Four different polymerization techniques they are described to polymerize vinyl type monomer, namely. So the first one is emulsion polymerization. So in emulsion polymerization is resulting in 0.05 to zero point 0.2 micrometers particles are present, and dispersion polymerizations resulting in 0.3 to 10 micrometers particles. While in precipitation polymerization, its result is nanospherical and its particle size will be between 0.1 to 10 micrometer particles. While in suspension polymerization, the resulting is 20 to 2000 micrometer particles. Now come toward the emulsion polymerization in continuous aqueous phase. So in this process, the continuous aqueous phase emulsion polymerization is one of the most widely used procedure to make the nanoparticles from drug delivery purpose, especially those prepared from the alkyl cyanoacrylate monomers. So an oil and water emulsion system is employed where the monomer is emulsified in a continuous aqueous phase containing soluble initiator and surfactant as well. Under these conditions, the monomer is partly solubilized in micelles 5 to 100 nanometers emulsified is a the monomer droplets 1 to 10 micrometer and partly dissolve in a continuous aqueous phase. The polymerization takes place in the outer continuous phase and not in the micelle interior. Initiation of polymerization occurs in the aqueous phase when a monomer molecule collides with an initiator molecule, which may be an anion or free radicals. Alternatively, the monomers can be struck by a high energy photon, such as gamma radiation and ultraviolet or intense visible light. So, propagation occurs when the nascent monomeric radical interact with other monomer molecule, forming primary particles. The polymerization is maintained by the continuous supply of monomers from the monomers reservoirs such as droplets and micelles, and they are illustrated in the figure 3. The surfactant also play a very significant role in the process. Important, which is surfactant is the important component of this process in which act to stabilize the growing polymeric particles by surface absorption. Phase separation and the formation of the solid particle occur before or after termination of the polymerization process. Polymerization can occur in some systems without the presence of surfactants. Various particulate systems have been prepared by these methods, including polystyrenes or polyvinyl pyridines and polyacrylines and polyglutaraldehyde Considerable work has been done using the polyalkyl cyanoacrylates PAC and nanospheres due to their biodegradability and bioelimination, so ease of preparation, low toxicity, and stability. Although initially used as homeostatic acids, aids, and surgery, their preparations was first recorded by the Carvers and, and their collaborators. Monomers was emulsified in a stirred acidic aqueous medium containing a non-ionic surfactants, which is also given in the figure 4. So the suspension obtained display a blue coloration due to scattering applied by the particle known as the Tyndall effect. Glucose, twin, dextrans 70 and Pluronics F68 have been used to stabilize and protect the particle from the agglomerations. Various monomers they are available such as methyl, ethyl, butyl and isohexyl monomer to prepare the PAC and nanoparticles which different in the length of their alkyl side chain. The polymerization is an anionic mechanism initiated by the hydroxide ions or any base present. The reaction scheme can be given in the figure 5. The polymerization rate is regulated by the hydroxyl ion concentrated in hence and the carried out at the pH value below 3.5. Above this pH, the reaction rate is too rapid to allow the discrete particle formations. This is a very simple presentation of the emulsion polymerization mechanisms in aqueous phase. This one you can see here, these are nanoparticles, and this one is monomer droplets. You can say these are the monomer droplets. And these are the growing polymer radicals, and this one is the dissolved monomers. Look at this shape. These are the monomers in micelles which form a network-like structure. You can see here in another sketch the preparations condition used in preparation of polyalkyl cyanoacrylate nanoparticles. This is mechanical stirrer, and this one is beaker, and this beaker you can see there is acidic polymerization medium containing surfactants as well as the nanosphere which is formed and display the Tyndall effect. This is the Tyndall, and these are the monomer which is added drop wise to uh, surfactant. The main advantages of this polymerization is that it can be carried out at room temperature, which is 25 to 30 degrees centigrade without energy input. PAC and nanoparticles have been prepared reproducibly at a semi-industrial level and can be made aseptically. Spherical particles of around 200 nanometer in diameters, they are prepared by this technique as demonstrated by the scanning electron microscopy. Particles as small as 10 nanometers were obtained by altering the concentration of surfactants and by increasing the amount of sulfur dioxide. Freeze fracture study confirmed that PAC and nanospheres have a dense solid spherical core with no distinguishable surroundings membranes. The content of polymerization's medium such as pH, concentration, type of surfactants and monomers type have been shown to influence the particle size. A large variety of drugs such as cytostatics and antibiotics have been encapsulated in PSC and nanosphere which successfully at the parts due to their porosity and high specific surface area. Drugs are incorporated into these nanosphere during the polymerizations or adsorbed onto the surface of the preformed particles. The loading of lipophilic drug is limited by their solubility in the aqueous acidic continuous phase. Acid labile or acid sensitive drugs can be incorporated using polydialkyl methyls malonate ester as they are polymerized at the neutral pH, and this is due to the presence of the less electrophile alkoxy carbonyl group as compared to the cyano group of the alkyl cyanocrylates. This use, however, has been limited due to their slow biodegradability. In emulsion polymerization in continuous organic phase, this procedure involves emulsifying a water-soluble monomer in a continuous organic phase containing a soluble initiator and surfactants. This constitutes a water and oil type of emulsion as the different phases are reversed from more common continuous aqueous phase based methods. The first process reported for the nanoparticles formation by the parents Beige and Spicers is the example of this procedure. Polyacrylic nanoparticles were prepared by dissolving the acrylamides and N-N-methylenes bisaccharylamides is an outer hexane phase containing anionic surfactants. The polymerization was initiated by the gamma ultraviolet or visible radiation. So biologically the active and antigenic material were encapsulated and show intact biological activity and high antibody productions after encapsulation. The spherical particles of 18 nanometers were produced based on electron microscope studies. Crowther reported that the transmission electron microscopy team revealed that the particle to have a solid dense interior forming a matrix-type nanopellets. Ekman and Jolom investigated the polymerization of those monomers by radicals, by the radical initiations and the Calvin chloroform mixture, but higher particle size were obtained. The main disadvantages in using the polyacrylamides systems and that are not biodegradables and the monomers are toxic. The extensive purification is also required to remove the organic solvents, anionic surfactants, and residual monomers. Erdmann and its collaborators produced the biodegradable polymers polyacryl dextrane particles by incorporating the dextrane into the polyacrylamide chains. In these particulate systems were metabolized and eliminated faster both in vivo and in vitro than polyacrylamide particles. Another one is dispersed polymerization. So in dispersed polymerization, the monomer and initiators are dissolved in the continuous phase which act as a non-solvent for the developing polymers. The continuous phase can be organic, aqueous, or the mixture of miscible phases. Two methods of initiation have been employed including the gamma radiation and chemical initiation by the potassium peroxide disulfide. As the polymer is formed, it precipitates as a nanoparticle, and these nanoparticles they are not polymeric precipitates as in precipitation's polymerization, rather they are swollen by a mixture of the monomers in the continuous phase. Crowther and Spices develop a dispersion polymerization producing adjuncts nanospheres of polymethyls and methacrylates PMMA. The monomers is dissolved in the phosphate buffer saline and initiated by the gamma radiations in the presence and the absence of influenza variants. This system shows enhanced the adjoint effect over the aluminum hydroxide and prolonged the antibody response. Polymethyl and methacrylates particles could be distinguished by the TEM studies, and the particle size was reported elsewhere as to be 130 nanometer by the proton correlation spectroscopy. The particle size could be reduced and producing the nonce or monodispersed particle by inclusions of the protective colloids such as proteins or casein. Polymethyl methacrylate nanoparticles are also prepared by chemical means using potassium's ferroxo disulfate as initiator combined with heating at 65-25 degrees centigrade. Increasing monomer concentration and temperature resulted in increase in particle size. Copolymerization of his beans carried out for the purpose of producing more hydrophilic particles, and the methyl methacrylate is being copolymerized with the methacrylic acid and ethylene glycol dimethacrylate and hydroxypropyl dimethacrylate or 2-hydroxy ethyls and methacrylates by emulsion polymerization. The particle sizes from 30 to 340 nanometer in diameters can be produced by chemical initiations, and zero point 0.3 to 3.0 micrometer particles produced using gamma radiations. Antibodies, amino acid, and fluorescent molecules can be covalently bonded to the active surfaces groups on these particles by cyanogen bromides, ANS, carbodimides, and glutaldehyde methods. The polymerization of PMMA and its copolymers avoid the use of organic solvent in inorganic surfactants. However, the PMMA is not biodegradable and thus persistent in the body. So the hydrolysis of these polymers required in acidic environment, not generally found in the body fluids. So once again, look at the preparation scheme for the emulsion evaporation methods. This is also the method which is used for the preparation of nanosphere nanoparticles. So this is mechanical stir, and this one is, you can see here, the same method which is applied here. This is aqueous continuous medium containing surfactants. This is also magnetic stirrer, while the same case is here, and there is evaporation under reduced pressure. So in first, and you can see in first semi-solid nano droplet that is formed. So here the polymer and drugs, here the polymers and drugs, they are dissolved in water miscible solvent, which containing the aqueous continuous medium containing surfactants, and which the semi nano droplets they are formed. You can see here. This is a mechanical stirrer. Look at the second case. There is the diffusion of the solvent from the nanodroplets particles. So the diffusion is take place here, and the solvent is being diffused from the nanodroplet particles. It should be under a very low pressure, so it can be evaporated under reduced pressure. While in third case, the solid nanoparticles, so they are formed. The solid nanoparticle is formed, so with the help of this mechanical stir. So there are different methods through which you can formulate and prepare the nanoparticles, nanospheres, by different processes like polymerizations and many more processes they are used. Inshallah, in my next lecture, we will talk about the nanospheres prepared from the preformed polymers. For example, you have preformed polymers, and from these preformed polymers, how the nanospheres they are prepared. Thank you so much for watching my lectures. So, if you need these contents, so they are already available with me. So, I will share with all my students, with all my viewers. So inshallah, in my next lecture, we will talk about the nanospheres preparation from the free-form polymers.